At least that's all it was.
I know.
It could have been
All
more.
right, so we're starting Friday. We talked about glycolysis. I changed some of our PowerPoints to indicate the new textbook. So in your book, this figure was in there. I wanted to show it. So this glycolysis, they color code blue throughout your whole chapter. So you kind of maybe can think about that when you do it. And then pyruvate oxidation and the Krebs cycle, which is also known as the citric acid cycle, is like the red brick color orange. And then oxidative phosphorylation, which includes the ETC and chemiosmosis through ATP synthase, is going to be purple. So this is the new figure that's in your book. So Friday we talked about glycolysis. Glycolysis was the splitting of glucose into two sugar molecules of pyruvate. Sorry, by the way, if my phone rings, I have to step out. It's because my gastroenterologist called me back and I have to talk to them. Anyways, we made a little bit of ATP through a process called substrate level phosphorylation. You need to know about that. It was 2 ATPs are synthesized per 1 glucose. You also made 2 NADH molecules per 1 glucose. Now, if there is no oxygen, then we don't go any further into the mitochondria. Okay, what has to happen is a process called fermentation. Got it? I'm going to go ahead and talk about fermentation right now. We didn't talk about it on Friday, really. So, fermentation happens when there's no oxygen available, and the only way that the cell has to make ATP is through using glycolysis. Okay? So, let me just move on. Some terminology you should know from before: aerobes and anaerobes. An aerobe is an organism or a cell that requires oxygen to live. Okay, it synthesizes ATP. There are things called obligate anaerobes or obligate aerobes. Means you're obligated to have oxygen. If you don't have it, you'll die. Okay. There's also something about that. Now, anaerobes are organisms that don't need oxygen. And there's obligate anaerobes, things that cannot have oxygen. One of the bacteria that can cause acne on your face, it lives in like thrives in the absence of oxygen when your pores are clogged. So one of the ways that the medications work is by cleaning your pores out, opening them up. When they're exposed to oxygen, it kills the bacteria because it's an obligate anaerobe.
Are there like animals that live in your hardware? I don't think it's dust mites. It's all sorts of mites. And it's like a bunch of like mites.
Have you seen an article that talks about how like sometimes it might be good not to make your bed because if you leave your covers exposed, it can let all that dry out because when you sleep, moisture gets in there and increase the growth. Great! I never tried that. Not like I do. Now she doesn't complain about them.
Don't clean my sheets.
Oh, wait, you should clean your all that red skin.
Wait, is it true that your bed double inside dust mites?
In math. Ow. Yeah, that hurt. Yeah. It will too. Yeah. Yeah. All right, anyways. There's facultative anaerobes that can live with or without oxygen, and those are the ones we're really going to talk about.
So there's no scientific view.
They're kind of the same thing. We just call them facultative anaerobes, but this is the same thing. Okay, I just talked about this. Obligate anaerobes. They can't function in the presence of oxygen because it's a poison or toxin to them. So some organisms, now, prokaryotes are very diverse. These are, remember, prokaryotes are single-celled, basically bacterial organisms, either archaea or prokaryote. Sorry. Anyways, sometimes they can actually use their own cell membranes as sort of like a modified ETC, and it's not within a mitochondria. Okay, and they can have enzymes that can basically kind of carry out a Krebs cycle-like thing. Okay. we'll talk about that later. But if they can do that, then they won't use oxygen as a final electron acceptor. They would use something else if they do that at all. Sometimes they just use glycolysis, yeah.
Is there a facultative anaerobe?
It's the same thing as a facultative anaerobe,
Oh,
because
okay.
they can use both.
Somebody else does.
Yeah, it's
Are
okay.
we?
It's okay. Well, we're really obligate because we can't survive for very long without it. Although our muscle cells can undergo lactic acid for just a little bit. Okay, so remember, this is the equation for glycolysis itself. And this is the way that cells make ATP without oxygen. So if an organism, if they're, let's say, a facultative anaerobe, and they can do both, well, what happens if there is no oxygen? Well, they'll just do glycolysis. But how do they keep going? As long as they have glucose still there, as long as they have ADP and phosphate so they can put them together, the reactants they would need. Okay, the cell will use up these ATPs pretty rapidly and make these again. As long as it has a food source, it'll have this readily available. But where is it going to get more NAD+? If the NADH normally would take the electrons and drop them off in ATP. If there's no oxygen, it's not going anywhere. It doesn't have anywhere to drop off its electrons. So what happens to it? How do we get that NAD+ back? We go through fermentation. Fermentation is the process of basically taking those electrons and the hydrogen off the NADH and regenerating the NAD+. That's the point of fermentation, is to regenerate the NAD+. So glycolysis can continue. And then you can make more ATP. Okay. So how is it recycled? Something has to accept the hydrogen and the electrons and hydrogen from the NADH, another molecule besides the ETC. All right. And depending on the type, you can have ethanol or lactic acid fermentation, and we'll talk about the two things. These are both in your book. You need to know them. So here is alcohol fermentation, and I'll have some videos to show you on these things today as well. So alcohol fermentation, I'll go back to that in just a second. So here are a couple of different types of bacteria that they undergo different types of fermentation. So alcohol fermentation or ethanol fermentation is here. So specific species of bacteria will undergo this. All right. We can use alcohol fermentation to our benefit because that's how you can make wine. Anyway, and beer, actually. Yeah. Yeah. And then the bread rising is through the carbon dioxide. Yeast undergoes alcohol fermentation. Is yeast a bacteria? No. What's yeast? A fungus, right? Yeast a fungus. So it'll do this, and we use it for its CO2 to help bread rise. Anyway.
What about like tofu or like yogurt?
Okay, so that's over here. Lactic acid fermentation, and then the soy sauce, and yeah.
Tofu is basically the curd of.
So let's do alcohol fermentation. Here we have glycolysis, the green area at the top. Right? In alcohol fermentation, what happens is that pyruvate, a CO2 is lost, so this CO2 that's highlighted in blue pops off. There it goes. It'll just diffuse out of the cell. And then it converts to something called acetaldehyde. And here, this acetaldehyde itself serves as the electron acceptor. An enzyme is here. That's not, they don't show it to you, but an enzyme takes the electrons and hydrogen off of the NADH, pops it on acetaldehyde. We regenerate the NAD+, so glycolysis continues, so we can make more ATP, and the byproduct is ethanol. And when I'm not feeling good, this becomes my diet. And maybe if I can eat that, maybe. Something called the brat diet. Bread rice applesauce. Huh?
Nice.
Oh,
It helps me. It's a way that I can get food without making it more upset than it already is. Have they talked to you about this?
yeah.
Yeah, okay.
Oh, yeah, I was gonna do the same.
Yeah, wait, what is already anyway?
Yeah.
You get that feeling where like you have to go, so you don't wait.
Yeah.
Oh.
That's what I eat when I like
Having that joy,
I
like,
know the
Right,
outside.
so no, right, no easy thing.
Food up my
You should eat stuff.
It's
I mean, I do anyway. It's like, oh, I know I shouldn't do it, but I'm not.
Shouldn't be what?
I'm not I had that all the time. I'm insane.
See, like, I'm normally in my bed, I'm right there.
Okay, sorry.
Where the food is, yeah, so.
I totally.
No,
I totally understand. I'm 3 weeks on medication.
I did.
So that'll be fun.
Probably.
Okay, so that.
Just when I'm trying to like text or something.
Yeah. Oh, yeah, Okay, that's that. Lactic acid fermentation. We do that in our skeletal muscle cells. It will not sustain you for very long, okay, at all. Because at some point your cells will undergo something called oxygen debt, and then you'll have to start breathing heavily to get that oxygen back in. You ever go up the stairs really fast, like in the school, and by the time you're like from the first floor, you're trying to replenish the oxygen debt.
That gives me to literally go up the stairs and I'm fine, and then I get up here and I'm like 2 feet down the hallway from the stairs.
That's like me going around the hallway, like.
You know you're.
Yeah. Anyways, some fungi can undergo lactic acid fermentation. We utilize this, or they use it to make dairy and yogurt. But the pyruvate itself serves as the electron acceptor. So an enzyme is there to just pop those electrons and hydrogen off, put it on pyruvate, you make lactate, and we regenerate the NAD+. The point of fermentation is only to make the NAD+, so that the cell can keep undergoing glycolysis to make the ATP, okay? Now what happens with the lactate? In skeletal muscle cells, what happens, y'all, a common sort of misconception is that the lactate is what causes you to be sore. Okay, it doesn't cause you to be sore because the lactate is taken out pretty rapidly and taken to your liver where it's converted back to pyruvate. Okay, what causes your muscles to be sore, like the micro tears that you put in them from working out? The lactic acid, if you're undergoing this process for a good amount of time, it'll drop the pH of your cells, and they might not work as hard, or that's what can cause some of the you won't be able to work as hard and things like that, and you can stop working. Yes.
So what about like if you squeeze something really fast and then all of a sudden it starts to hurt more? Is that like?
It could be the pH is dropping. I mean, it could cause a little bit of that, but like the next day, people talk about you're not sore the next day because the lactic acid is still there.
Is that like what the milk sickness does to your body?
Kind of, yeah.
What about that masochism thing?
Oh, that's not.
Do you
Yeah. All the myths.
Oh, I know.
Spread knowledge.
Good, it's what should happen. Some people can train in this Michael Phelps would train in this anaerobic zone where he would train and try to increase his lactic acid production because, if you swim underwater, you're not getting the oxygen. And the longer you could sustain that, you can save yourself a little bit of time, as long as you can keep going fast. So part of the way he would train, and sometimes people still train, is try to train in that zone to let your cells be used to working under that lower pH area and increase the conversion of lactate back to pyruvate. Anyways. So there's training that athletes do in this area.
It's something like when you push yourself on the way to the end without going up the air.
Anyway, what's interesting is that there's other types of bacteria that undergo different types of fermentation, and there can be different products. So notice over here, there's formic acid, all kinds of other different things. There's different types of fermentation, but you need to know the alcohol and the ethanol. This was just kind of interesting. I put it on here because we use different fermentation processes to produce different things for our own use. So beer, wine, fuel. What you do for fuel is you start with particular agricultural waste. You can use Saccharomyces cerevisiae. Anyway.
What does Swiss cheese have to do?
Swiss cheese?
Why is Swiss cheese?
Why is it?
Why is it different?
It has different end products, so you make some sort of.
Oh, that's why it's got
acid.
holes in it. So
Yeah.
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 Oh.
Wow.
Oh my goodness. I know.
Exactly.
It's so fun. Sauerkraut. So I think Kenny's like research project based on that. Yeah.
I feel we made it feel summer fall. We like made him sit down. And he like destroyed it in terms of class. From read like 20.
Mr. Sabla should have done it in research one before he got to me. Well, see, I'm just saying. That's not my problem. Just my job is to make y'all better.
He like destroyed his day.
Depending on where you start from.
Destroyed his whole life this fight. He's never gonna be a PhD scientist now. Like.
Oh, anyways, okay. That's all. Notice, flavoring, different types of flavorings. Vitamin C.
You know what's
Look at that summer sausage on it.
15
All right,
like,
I have a video about beer. Yep. And then we'll move on.
yeah, fresh octopus. Okay.
What's it? Fresh octopus?
What?
Fresh octopus.
All right, listen, I have 2 videos. Enjoy these before we start talking about the Krebs cycle.
Cider, that's like hard cider, not just juice.
Or anywhere else other than America.
21 years old and you can have some of this, okay? Okay.
They teach us how to make
We're going to a backwoods speaking.
Don't think I'm gonna go in my country.
Very, very dangerous. Very unsafe. Different rule of cider. We're going to get freshly pressed sweet cider. Add a specific type of yeast to it, a little bit of sugar, tap it off to prevent any other contaminants from getting in there, and just leave it to sit for a couple months, and we'll end up with some good booze. So what I'm starting with is this easy to carry jug of pressed apple cider. This is a sweet cider, meaning there's no alcohol in it. So what happened is we just took a whole bunch of apples with the skin, seeds, everything on them, pressed them, juiced them, and ended up with this. This is going to be my starting material to make a really good, strong cider. I'm going to pour this into the fermenter, and I'm going to add some brown sugar to it. You can use any different type of sugar, but it is good to add a little bit, especially in the beginning parts, because yeast's favorite thing to eat is glucose. And there's easily available glucose in these simple sugars. I use a brown sugar because I like the flavor of it a little bit more, and the more that I add, the higher the alcohol is going to go up. So if I added this whole bag, I might end up with 14% alcohol, something around there, which is really high. It's going to taste terrible. So instead, I'm just going to give it a little Kickstarter with some brown sugar. And I'm also going to add a little bit of yeast nutrient in there, just to give it that initial kick to get the yeast to really start blossoming and reproducing. And then it's eventually going to move on and start breaking down the fructose in here to make our alcohol. Forget the you. So just so you know, I dump this in, I dump that in, we mix it up, and we're done.
Oh man, this is such a short
And
video.
we have booze.
And that's it?
That's it. So with my cider, I'm not too strict about measuring anything. I'm not looking for complete reproducibility. I kind of like the fact that I'm going to get a little bit of a different flavor profile each time that I make a batch. So I use all different varieties of apples, all different varieties of yeast, different types of sugar, and I kind of experiment, and just sometimes I make some really great stuff. Sometimes it's not so great, but it all has booze in it.
All right, now let's talk about the science involved in this process. Converting this juice into alcohol starts with yeast,
Okay, listen.
a fungi with the scientific name Saccharomyces cerevisiae. Now inside the cell, glucose molecules, think sugar here, are broken down via glycolysis into two pyruvate molecules. Then to complete the process known as alcoholic fermentation, those two molecules are broken down into two molecules of carbon dioxide and two molecules of acetaldehyde, which are then converted to ethanol. Basically, the concentration of ethanol is the concentration of alcohol in the final mix. And it's also important to note that there are many species of yeast and many variations of brewing yeast, which is Saccharomyces cerevisiae. Choosing the right one changes the final mix.
You can use a specific cider yeast, but that produces kind of a sulfur smell, which is just really nasty, especially if you're brewing it in your house. You can use an ale yeast. You can use different wine and champagne yeast. A lot of people like to use an English beer. I don't think that works too good. The champagne is going to produce a very, very dry cider, so it's going to use all of the sweetness from it, and that's where a non-fermentable sugar comes in. So if you want to back sweeten a really dry cider, so add some sweetness to it, but you don't want the yeast in there to eat the sugar and to convert it to alcohol, so then you get more booze but less sweetness, use a non-fermentable sugar like lactose. Yeast is actually lactose intolerant. It lacks the enzyme that's required to break lactose down into energy and produce alcohol as a waste product. So you add some lactose to it, it adds sweetness that the yeast can't use. So you pop the airlock on, then get cider, shake it up a little. You get that yeast nice and mixed up, make sure it can access all those sugars. Just leave it to sit for a couple weeks. Oh, look at that. Let the yeast do its thing, which is break down those sugars into alcohol and carbon dioxide. And then, probably about 3 weeks from now or so, once I see that it stops bubbling, take it out, we've got some good cider. Now at that point it's going to be still, so non-sparkling. It's just going to be a flat alcoholic cider. So what you can do then, there's still going to be active yeast in there. So I can transfer it over to another jug, add a little bit more sugar, and I'm going to use a really easily fermentable sugar this time. So I wanted to use a slightly difficult to ferment sugar to let it go a little longer, but to carbonate it, I'm going to use a really easy to ferment sugar, something like corn sugar, or just straight white sugar right out of the pantry, and just a small amount of it. For a gallon, I'm probably only going to use about 2 tablespoons. I'm going to put that in, and then I'm going to bottle it. So as soon as I add that fermentable sugar, I'm going to put it into bottles like this and cap it, and then that closes the system. So now you've got yeast in here that's eating those sugars and producing carbon dioxide, and that carbon dioxide is going to carbonate your cider, and then you get a nice sparkling.
I learned something, I hope you did too. It's kind of how to, but a little bit of science mixed in. So it was my first time to work with Pat here in Boston, which was really fun. And if you want to learn more about what Pat does, well, I'm going to let him explain.
Thanks very much, Rob. Yeah, this was awesome. So if you want to check out some other things that I do, you can check out the link right here, which is naturecalls.tv. See some of the insane things that I do with my life. A little bit more animals, a little less alcohol. And if you go to the description of this video, you'll find some other great links to some cool stuff that we've done. So thank you very much for watching, and enjoy your cider.
If you guys haven't figured it out yet, Pat's a little bit more nerdy than me. I just think I'm a little, I'm a little bit cooler. We were trying really hard to make him look cool. If you just see his picture, he looks almost too cool for school, but he's actually in there. He fits in with the group. Like I've seen in that video.
It's fun to be a nerdy scientist. All right, next.
Flatulence, or passing gas, is a normal daily phenomenon. Most individuals, yes, that includes you, will make anywhere from 500 to 1,500 milliliters of gas and can pass gas 10 to 20 times a day. But where does this bodily gas come from? A small proportion may come from injecting air during heat or at other times, but the majority of gas is produced by bacteria in our intestines as they digest parts of food which we cannot. Our intestine is home to trillions of bacteria living in a symbiotic relationship with us. We provide them with a safe place to stay and food to eat. In exchange, they help us extract energy from our food, make vitamins for us like vitamin B and K, boost our immune system, and play an important role in gastrointestinal barrier function, motility, and the development of various organ systems. Clearly, it's in our best interest to keep these bacteria happy. Gut bacteria get their nutrition primarily from undigested food, such as carbohydrates and proteins, which come to the large intestine. They ferment this undigested food to produce a wide range of compounds, such as short-chain fatty acids, and of course, gases. Hydrogen and carbon dioxide are the most common gaseous products of bacterial fermentation and are odorless. Some people also produce methane due to specific microbes present in their gut, but methane is actually odorless too. Well then, what stinks? The foul smell is usually due to volatile sulfur compounds, such as hydrogen sulfide and methanethiol, or methyl mercaptan. These gases, however, constitute less than 1% of volume and are often seen with ingestion of amino acids containing sulfur, which may explain the foul smell of gas from certain high-protein diets. Increased passage of gas is commonly noticed after eating foods with high amounts of indigestible carbohydrates, like beans, lentils, dairy products, onions, garlic, leeks, radishes, potatoes, oats, wheat, cauliflower, broccoli, cabbage, and Brussels sprouts. Humans lack the enzymes, so the bacteria able to ferment complex carbohydrates take over, and this naturally leads to more gas than usual. But if you feel uncomfortable, bloated, or visibly distended, this may indicate impaired movement of gas along the gastrointestinal tract. It's important not to just blame certain foods for gas and bloating and then avoid them. You don't want to starve the bacteria that digest these complex carbohydrates, or they'll have to start eating the sugars in the mucus lining of your intestines. Your personal gas will vary based on what you eat and what bacteria are in your gut. For example, from the same starting sugar, the bacteria Clostridium produces carbon dioxide, butyrate, and hydrogen, while Propionibacterium can produce carbon dioxide, propionate, and acetate. At the same time, methanogens can use hydrogen and carbon dioxide produced by other bacteria to generate methane, which can reduce the total volume of gas by using up hydrogen and carbon dioxide. So there's a complex web among intestinal bacteria, allowing them to flourish by either directly consuming undigested food or using what other bacteria produce. This interaction largely determines the amount and type of gas produced. So gas production is a sign that your gut bacteria are at work. But in some instances, people may develop abnormal increased flatulence. A common example is lactose intolerance. Most individuals have the enzyme for breaking down lactose, a sugar present in milk and milk-derived products. But some people either lack it entirely or have a reduced amount, such as after a gastrointestinal infection. So they're unable to digest lactose products, and they experience cramping, along with increased flatulence due to bacterial fermentation. But remember, most gas is produced as a natural result of bacterial fermentation in the intestine and indicates healthy functioning of the gut. The amount and type can vary based on your diet and the bacteria in your intestine. Exercise social courtesy while passing gas, and do try to forgive your bacteria. They're only trying to be helpful.
What about then when they order lots when they're having a heart attack?
What?
You know? When
No.
And they have like increased flatulence?
No.
Yeah.
What?
It's like you're talking about like myth busted.
If you have a heart attack, you'll have increased flatulence.
No, I mean sometimes it's like a symptom beforehand that they like end up finding a lot and they like have like
It's like a possible indicator for
yeah, I swear because like I read when I read Hatchet we were talking about that and then we're in like Parks and Rec.
Heart disease.
Yeah,
Health articles
it's
using
heart
that
disease.
Well,
Heart disease can have heart attack.
but like and like
If you have like a really bad heart, you're not gonna.
in Parks and Rec too, Jerry's like Parks.
Okay, come on. No.
No, I'm serious. Like I
Let's look. I've never heard that before, ever. No.
I have a quick question.
Yes.
What you call it? How come alcohol is banned till 21, but cigarettes are allowed?
Don't ask me those questions. I don't know that.
Well, mine are soft. I don't know. Yeah.
I've never heard.
Oh, it's a ridiculous myth. Okay, that's fine. I'm sure you
Well, I don't know. I've never heard that before. All right, we're gonna move on. I don't know. Hold on a second. Oh, that's Michelle Spagner. Okay, I don't care. All right, ready? That wasn't my doctor. Okay. So after we make our pyruvate, assuming there is oxygen present, and we're in a eukaryotic cell, we have mitochondria, then what has to happen is that the pyruvate moves into the mitochondria. We undergo pyruvate oxidation, and then the citric acid cycles, and then eventually oxidative phosphorylation. So right now we're going to talk about the pyruvate oxidation and the citric acid cycle. So again, glucose, when we started, we had a six-carbon molecule. It went through a series of 10 reactions or 10 steps in glycolysis, and it slowly was converted from glucose into a 2,3 carbon sugars called pyruvate. The first 5 steps were the energy investment phase. We used 2 ATPs. The second 5 steps, we produced 2 NADHs and we produced 4 ATPs. So the net total ATP production was 2, right? Okay. So here we go. At the end, we have 2 ATPs that can be used by the cell to do whatever. 2 NADHs that can go to the ETC, and then we have 2 pyruvates. So I'll turn that down. We always have to count our carbon. All right. So there's a lot more energy, a lot more electrons in pyruvate that can be taken off. So we still have a lot more to oxidize. So if, like I said, oxygen's there, then pyruvate will go into the mitochondria. And eventually all those carbons are lost as CO2. All of them. All right, so remember the mitochondria is a double membrane structure. The outer membrane is smooth. The inner membrane is highly enfolded. It has, the folds are called cristae. The inside of the inner membrane is a fluid called the matrix. In the matrix where the mitochondrial DNA is located, and there's ribosomes, and the proteins for the Krebs cycle are inside the matrix. The proteins for the ETC are actually in the inner mitochondrial membrane itself. And then the space between the outer and the inner membrane, is just called the inner membrane space. It's real creative. And that's what it actually looks like using a SEM, or transmission electron microscope picture. This is like a long ways, and this is one as well, but just taken from like the other angle. We learned all this already, right? Okay, this is all just review. What kinds of cells in your body are going to have lots of mitochondria? Muscle and neurons. Thank you. Okay. So remember, mitochondria, through the endosymbiotic theory we talked about, mitochondria's ancestors were free-living bacteria, and they show similarities to themselves. They're what we call semi-autonomous within the cell, so they will divide on their own within the cell. They'll move around in their own in the cell. They're not part of the endomembrane system. Same for chloroplasts. Anyway, okay, we talked about all this. So here's our mitochondria, the outer membrane, the inner membrane. The matrix would be in here. This is the inner membrane space out here. The pyruvate will go in to the mitochondria, and the first thing that has to happen is something called pyruvate oxidation, where it's converted into something called acetyl-CoA. Okay, this pyruvate oxidation step is the second step of the whole aerobic cell respiration. So the first thing that has to happen is the CO2 pops off. And so acetyl-CoA is only 2 carbon compound. There goes our CO2. NAD is reduced to form NADH. Yay. Again, that will take the electrons to the ETC. And then acetyl-CoA is produced. How many times does this happen for one glucose? Twice, because there's 2 pyruvates. Good. So for one glucose, in this step here, we make 2 NADHs. We produce 2 CO2s. What happens to the CO2? It'll just diffuse out of the mitochondria and out of the cell. Right? Okay.
What does the CoA stand for again?
Coenzyme A.
After eating oil.
Yeah.
Of all the things, remember? I remember the synaptic neuron. Of all the things to remember, I'm just saying. Of all of the things.
Remember what you taught us in the last 3 weeks of school? We didn't really
Oh, well.
Here's
Here's the problem with anatomy and research. At the same
like, I'm
time.
gonna start teaching on the opposite of the class, so you guys don't worry.
All right, pyruvate, here we go. Pyruvate is highly hydrophilic. All right, so it has to move through membrane proteins. Just remember that. I just want to show you that it's there. So this is showing stepwise. It's kind of like the picture in your book. The CO2, this CO2 right here. Notice down here, this is the one that's already been fully oxidized. There's no more hydrogens left over here. So this part pops off, and there we go. And then the NAD becomes reduced to make NADH. And then coenzyme A is attached down here. And then we have our acetyl-CoA. There's still hydrogens left with their electrons. Yeah. So there's still stuff in there that can be taken off. So that was our little oxidation step. Okay. So the next thing that has to happen is that acetyl-CoA moves into something called the citric acid cycle, also known as the Krebs cycle. Hans Krebs is the guy that discovered this. So therefore we named it after him. It occurs in the mitochondrial matrix. There's lots of enzymes that take place or that are utilized for this. It's called a cycle because it's a cycle. The starting reactant is the same thing as the ending reactant. It just keeps going and going and going. It regenerates the ending reactant every time. There's 8 steps in this pathway. Each step is catalyzed by their specific enzyme. You don't need to memorize them. The point of the citric acid cycle is to slowly complete the oxidation of glucose. The point of the citric acid cycle is to make those electron carriers. Those two go together. Complete the oxidation of glucose that we started with at the very beginning. Okay, it's acetyl-CoA now, and then to make electron carriers. The point is to make electron carriers. We make a little bit of ATP here. It's really negligible. It's not a lot. But the major point, we make a ton of electron carriers. A ton. It evolved after glycolysis because it will not happen when there's no oxygen. There was no oxygen on the early Earth. So, yes, it makes sense. I think I heard, I need to look it up when we talk about this stuff in the evolution of life, but I feel like I've heard something that oxygen probably evolved a lot earlier than they thought. Did anybody hear anything about that? No. Never. I think I was listening to NPR and I heard it on NPR. Tomorrow, during like second period.
Yeah, look into it, right? Do it.
All right, y'all ready? So, this is obviously not the diagram in your book. This is a one simplified, highly simplified. We're counting the carbons. Listen, yes. All right, so we have our 3-carbon pyruvate that we converted to acetyl-CoA. Acetyl-CoA will enter into the Krebs cycle. It immediately will bind to a 4-carbon compound. To make it how many carbon compounds? 4 plus 2 is 6. Great. All right, that is called citrate. You do need to know a few of these reactants and products in this pathway. All right. So 4 of these carbons in citrate are going to be regenerated to make this thing called oxaloacetate. 2 of the carbons are going to be fully oxidized and they'll pop off. So notice in this first step here, we go from 6 carbons to 6 carbons. Nothing is really happening in terms of we're moving into isomer. But notice we go 6 to 5. So we lose the carbon dioxide, and down here we go 5 to 4. We lose another carbon dioxide. So of the 2 we started with, we've just lost them both. There they go. All right. So the next few steps in the citric acid cycle in terms of the carbons, we're just reorganizing them and regenerating the end product with the reactant for the next cycle. You see that? Great. That's what's going on with the carbons. This happens 2 times because for 1 glucose we had 2 pyruvates, therefore 2 acetyl-CoAs. So how many CO2s are produced directly by the citric acid cycle for 1 glucose? 4. All this accounting, the numbering that I'm talking about, you need to be able to understand that. Because on the test I'll be like, how many CO2s are produced for like 4 glucoses? Okay, you'll have to know what you make for 1, you'll figure that stuff out. All right, now let's count the electron carriers that are produced. So during this process, we are oxidizing the acetyl-CoA or the citrate, and we're reducing these electron carriers. So at the same time we make our carbon dioxide over here, we're also producing some NADH. Same thing down here. Now over here we make a little bit of ATP. That's not an electron carrier. It's just a tiny bit. We make 1 ATP per acetyl-CoA. So 2 ATPs here for 1 glucose. If you were here Friday, you remember what I said about substrate level phosphorylation. That's how the ATP is made here as well. Yes.
What did you say?
Thursday, sorry.
What did you say was oxidizing?
The acetyl-CoA, but citrate. Thursday, I'm talking about Thursday then. All the notes, all that, yeah. Listen? But the same thing on the other side. When you have your next day off, y'all, I'm excited we're actually going to be here for a full week. Actually get stuff done.
Should we go to Boston? Next week, like this weekend.
Oh, yeah.
Wait, when are you going to be gone Monday?
No, we're gone Tuesday.
Oh, no. Yes, I know.
Yeah, Saturday.
That's fun. I love softball. It's like a million boys.
It's like 1 year girl.
Oh, yeah.
Don't get left behind.
And there are a million boys.
Yeah, she got left behind last year. What? Yeah. They all know where.
Yeah, they like.
I love
You need to get on the seat when you're supposed to get on the seat. Right. Anyway, okay. You have to come back. Alright, we make a little bit of FADH2. Alright, FADH2 is an interesting electron carrier. The hydrogens it's carrying are more like 7-year-olds. They don't have that much energy compared to the 2-year-old ones that the NADH are carrying, okay? They're a little bit lower energy. And we'll talk about that a little bit later. But the point is that they won't, in the end, they won't produce as much ATP as the electrons from NADH will. Anyways, we also make a little more NADH. So notice, per one acetyl-CoA, in the citric acid cycle, we make 3 NADHs and we make 1 FADH2. There should be a 2 on there, there's not. Okay? So for 1 glucose, we make 6 NADHs and 2 FADH2s and 2 ATPs. You see, there's a lot, a huge number. That's the point of this, is to make all those electron carriers.
You said 6 NADH and 2
FADH2s for 1 glucose.
Wait, is it FADH like 2, like subscript?
H2, yeah, subscript.
So for each like round, you make 3 NADH and then FADH.
Yeah. Alright, so this is actually the one in your book. Alright, so I'm going to show it to you. It kind of helps a little bit when you do your drawings to look at your book. This is why I'm saying you should bring your book or bring your Chromebook so you can look at it on there. Because it's interesting, they kind of color code some of these carbons. It makes it a little bit easier to understand if you're like me and you're like, well, how did that really attach to that? Where did that go and what happened? The blue carbons are the ones, notice here, just notice the blue carbons and then the red carbons. The red carbons are ones from acetyl-CoA that we attached. Here's oxaloacetate, that's how you spell that. There we attached our acetyl-CoA ones there. And this is a citrate molecule. Anyway, so in this first step, water comes out and goes back in. All you're doing is reorganizing, you're making isomers, isocitrate. You don't need to know isocitrate, but I'm just telling you, for those of you that need to know this. Notice in this step here, we lose our carbon dioxide. Now notice the CO2 that we lost is actually the one from oxaloacetate we started with, so that's okay. And then, but so we still have a 5-carbon compound here called alpha ketoglutarate, that's how you say that. And then we use our NAD plus and we reduce it to make NADH. Over here, the same kind of thing happens. The other CO2 pops off and we reduce NAD again to make NADH, and we have this thing called succinyl-CoA. This random coenzyme A with the SH group got added on. I'm just showing you there in this step. Now over here, this coenzyme A sulfhydryl group pops off, and then also notice, what do we make actually initially? We don't make ATP directly. What do we make? GTP. GTP is also an energy carrier molecule used by the cell, just not as much as ATP. Sometimes GTP actually will be used and the ATP won't be produced at all here, but sometimes it will be produced.
Where's the cheese?
It's a guanine. It's the same kind of thing that's used in to make DNA. What do you mean, where does it come from?
So confused.
Don't be confused. This is what happens.
So all we need to know is like what things to do.
Yeah,
We don't have to know like.
the enzymes or the actual things. No, the ones I want you to know are oxaloacetate and citrate. You don't need to know all these other ones. Okay. And you don't need to know their structures. You just need to know their names and how many carbons they have.
All right.
What is oxalate?
It's the end product of the citric acid cycle, and it's what the acetyl-CoA is bonded to make the citrate.
So it's the 2C,
That's the 4C.
the original.
There's 4 carbons.
The 4C.
Acetyl-CoA is the 2C.
Acetyl-CoA.
I wrote it down.
All right, and then notice we make our FADH2 over here. We add a water, and then we make our last NADH. You don't need to know any of these, I'm just saying. All right, so the citric acid cycle is called a cycle because it regenerates the oxaloacetate every time. So we can bond acetyl-CoA to make citrate. Yes. All right, so I'm not going to end. There we go. Okay, so at the end of this, glucose has been fully oxidized. There's nothing left. What is all we have left of the glucose? The electrons, those high energy 2-year-olds and a couple of 7-year-olds, okay? So we have to use our energy to make a lot more ATP, which is what we're going to do next. All we have at this point is 4 ATPs, maybe, not very much. So if we combine the citric acid cycle with that pyruvate oxidation step, if you combine them all together, okay, we made a lot of extra electron carriers. We made a total of 6 plus 2, so 8 NADHs if we combine it with that pre-oxidation step, yes? Okay, then we made 2 FADH2s. We made a lot. That's a lot of electron carriers that are going to the ETC. We'll talk about that later. So this is putting it all together. This includes the pyruvate oxidation step. Okay, so don't get confused. It's pyruvate oxidation plus the citric acid cycle. We made 2 ATPs, 8 NADHs, and 2 FADH2s. So if we start from a glucose, how many total NADHs do we have, including glycolysis? 10. 10. Because we made 2 NADHs and glycolysis as well. It's okay. You'll know this by the end of the day on Wednesday. You better. So.
Do you put in that name? It's the website for
So like, not
Why not? It's expected right now. Okay. You didn't have to know all these details in Bio 1. So, sorry. Yeah.
You said that the net gain was the Krebs cycle as well as the pyruvate.
These numbers here?
Yeah.
Yeah.
Okay.
That's where they were produced. So the pyruvate oxidation step plus the Krebs cycle. All right, so the value, I said this earlier, I'm going to say it again. The point or the value of the Krebs cycle is to make all those electron carriers. That's the point of it, to extract all those high energy electrons and put them in those electron carriers. That was the point. Okay, let me go to our next PowerPoint. Yep. Do what? Yeah. Are y'all ready? Y'all ready?
Yes. Is there a lot?
Is it ready? We need to do our drawing golf class tomorrow.
Is there a lot in the electron transport chain?
I have 26 slides.
All right. New page it is.
Okay. You ready?
Yes. This is
Okay.
4.
We made a little ATP so far, not so much. Okay. For glycolysis, we made 2 ATPs. We also made 2 NADHs. Krebs cycle, we made 2 ATPs. We made a lot of 8 NADHs if we include the oxidation step, and 2 FADH2s. But we need a lot more than 4 ATPs to run our body. 4 is not a lot.
10 million ATPs.
Yeah.
Oh, what the heck?
No, that game is part of the point if we're including glycolysis.
Millions and millions of times a second. Like instantaneously 30 years.
Alright. So your muscles need over 10 million ATPs per second. You can't make that many using just 4.
Wait, per muscle?
Yeah.
Wait, for like muscle cell
Good lord.
or in total?
Per muscle cell. Think about all those actin-myosin across the think about it.
No, like in total, there's one muscle cell.
But think about in one muscle how many myofibrils there are. And they're so long. Alright, listen. Have a hush over there. This is not easy stuff. Alright. So,
Wait, what are they?
nothing. They're just talking.
No, they're like shocked about how it has to happen.
Oh, it's crazy. I know.
We weren't talking.
It's okay. You're just being shocked. It's fine. I just got to move on. I want to finish the note taking in the class anyway. So you'll have all day tomorrow to start drawing, making your beautiful stuff, and cutting and making little movable electrons and stuff. All right. So the ETC is basically a bunch of proteins built into the inner mitochondrial membrane, along the cristae, the folds. And these enzymes and molecules are a bunch of proteins and enzymes all together. They're called cytochromes. And the point of the ETC is just to make a hydrogen ion gradient. That's all. Do you make any ATP in the ETC? No. The point is just to make a hydrogen ion gradient. Eventually, that gradient will be used via a process called chemiosmosis through something called ATP synthase to make ATP. We'll get to that in a little bit. Okay. Now, again, the mitochondria, here's our membrane. The ETC is found in here where I put that circle in the inner mitochondrial membrane all over the place. The reason they're all those folds is to have a huge amount of surface area. So you can maximize the amount of ETC proteins that you can possibly have. So let's look at it. This is kind of what the ETC would look like, just right here. Okay, I'll circle it. The thing on the end over here, ATP synthase, is not part of the ETC at all. And that is the ETC. The electron transport chain consists of a series of protein complexes. Y'all, that number 1 right there is a series of complexes. It's not just one giant enzyme. Okay? So 1, 3, and 4 are all integral membrane proteins. They go all the way through. Protein complex number 2 doesn't go all the way through the membrane. There's also some mobile little electron carriers. It's called cytochrome, or ubiquinone Q and cytochrome C. The little Q is totally hydrophobic. You see how it's in the middle of those, the little tails, the fatty acid tails? It's a completely hydrophobic carrier molecule. It fluctuates back between number 1, number 2, all the way to 3. So it picks up electrons from 1, picks them up from 2, and takes them over to number 3. Like a little shuttle bus.
So how does it get to number 2?
It just goes down. It's in the membrane. It just doesn't go all the way through the membrane.
Okay.
It just goes down. And then cytochrome C is a little peripheral protein on the outside of the inner membrane. It's in the inner membrane space over here, which is the orange. And it just takes electrons from complex 3 to complex 4. Okay? The yellow lines is basically, follow the yellow thread. The yellow lines are the path of the electrons. That's the way they go. The electrons can be dropped off in 2 places depending on where they're coming from. The electrons from NADH have the most energy, so they're dropped off at complex 1. The electrons from FADH2 don't have as much energy, so they're dropped off at complex 2. Once there, either way, they go to ubiquinone Q. They travel to complex 3, and then they go to cytochrome C, travel to complex 4. Eventually, they end up combining with hydrogen ions and oxygen gas to make water. Everything that I'm saying, you need to know. Everything on this diagram, you need to know. This is the diagram in the book of all, like glycolysis, you don't need to know all that. Krebs cycle, you need to know all this. I don't recall. No, 9 point something. All right. Is this overwhelming? It shouldn't be. All right. Anyway, hush. Now, there's a lot to this. You've got to focus and listen. Okay? So, we talked about these electron carriers, where they came from. So I'm going to focus on NADH right now. This diagram's a little bit different, but I'm going to focus on it. So notice, NADH, when it drops off the electron, okay, what happens is that the hydrogen, notice, the little extra electron pops into here, and then each hydrogen's carrying an electron as well. So the hydrogen itself drops its electron off and becomes a hydrogen ion. Okay, over here in the mitochondrial matrix. So there's 2 electrons that were dropped off for 1 NADH. 2 electrons are dropped off. Okay?
Same electron though, isn't it?
No.
No?
NAD plus is plus.
Okay.
You have 1 electron to like negate the plus to just become NAD period.
Okay.
And then the second electron is with the H. Does that make sense?
Yeah.
Does that make sense to y'all?
Yes.
Okay. So it dropped off 2 electrons. Where does that NAD plus go now?
Go back to
It can either go to glycolysis or just stays in the matrix and goes to the Krebs cycle. It's right there. Right, okay, good. Good. So, here's our electrons. Now these electrons, they have a lot of energy, okay? But as they pass through the ETC, they're losing energy. As they lose energy, the energy lost is used to pump hydrogen ions from the matrix into the intermembrane space. To actively transport hydrogen ions from the matrix into the intermembrane space. Yeah, all the stuff I'm writing is not written in here anywhere. Hopefully you should be taking notes or something unless you remember all this stuff. As the electrons move through the electron transport chain, they lose energy. And the energy that they lose as they pass through complex 1, complex 3, and complex 4. Do you see these blue arrows? Those blue arrows are there showing that hydrogen ions are pumped from the matrix into the intermembrane space.
Oh, it's a great agreement.
Yeah. That's how the gradient is created. It's actively transporting them. That means it's going against their gradient. So what's the pH like in this intermembrane space compared to the matrix? The pH is very acidic in the intermembrane space. There's a lot of hydrogen ions there. The hydrogens don't want to go that way in terms of the laws of thermodynamics. They don't want to go that way. Yeah. Okay. The electrons from FADH2, like I said, they're not as high energy, so they have to be dropped off at a lower energy level, which is this complex 2 over here. And so notice the electrons from FADH2 will only pump 2/3 as many hydrogen ions out compared to the number of hydrogen ions that will be pumped from the electrons from NADH. You see that? Because they only pass through 2 of these big complexes to pump out hydrogen ions, not the first one. So in the end, the electrons from FADH2 won't contribute to the production of as much ATP. At the end, remember the most electronegative element we know about? Oxygen. It has a really strong pull on electrons. That oxygen, it's like Halloween night. My kids are so freaking excited about Halloween tonight. They're like, candy. Okay, the oxygen is hanging out down there. It's like the guy with a giant thing of candy. And these little electrons are like my kids. They just want to get there. So the oxygen hangs out at the end of the ETC, and it helps to pull the electrons through it. It's like the bag of candy at the end of your trick-or-treat night or something is what you get, okay? Pulling them through. The oxygen combines with hydrogen ions and those electrons to make water.
So that's just the last complex.
Yeah, this is complex 4 right here. Yes. The very bottom part of this, notice it's called cytochrome C oxidase complex. Complex is an enzymatic, has enzymatic activity down here to catalyze this reaction. I'm going to show you a bunch of videos on all this, y'all. This is highly complex, I know, and you're like, whoa. I know. I've been doing this, teaching this for 14 years. So if you seem like, why does she know it so well? Well, it's my job, okay? So it takes a long time to get this. It's not completely obvious. So the point of this, y'all, is you need to understand the point of this to make the hydrogen ion gradient. That's the point of the ETC, to make that gradient. That's the point of it. Okay? And what powers these proton pumps? These are called proton pumps. It's the energy from the electrons they're losing that powers them. Yeah. That's why we have the electrons. They're losing their energy, and the energy they're losing is used by them to pump the hydrogen ion. One electron will pump out 3 hydrogen ions. One electron from NADH. There's 2 from FADH2. It's just kind of showing their path. Yeah. Because it has 2 hydrogens.
So is that like, it's not necessarily, is that the hydrogen that's being produced, or that's just being put?
Okay, well, either. There's actually a lot more that are there. Some of the hydrogen are made by these becoming oxidized, or the ones that are pumped out, but there's a lot of other ones. Because they're diffusing back in through ATP synthase as well. So.
that one electron from ADH comes out through what? It comes out through hydrogen ions. Yeah, it's okay. You like just had to start to slow down. But I have a quick question. So the electron is released from the from complex 4 back into the matrix?
Yeah.
To
It combines with these two hydrogen ions
okay.
and an O to make water.
For it to be
All right,
like
cool.
combined with the hydrogen.
Oh,
And the oxygen to make water. Okay, y'all listen. Y'all, okay. I'll point it out later. I said it like 4 times, but y'all know. Look, to make water, we need H2O, yep. There's hydrogen ions. In order to make them not an ion, we have to get an extra electron. So there's one electron makes one of these, just a plain H. Another makes it another plain H. We combine it with an O to make water. Yep.
How does the electron lose energy?
Okay, class is coming up. Hold on. I said all this a little bit, so it's in there. Hold on. Let me show you this picture. So remember how we talked about free energy?
Mm-hmm.
These are the different protein complexes, 1, 2, 3, and 4. Okay? If you look at their free energy that they have, you start out really high and you go lower. So as the electrons are being moved through these complexes, they're going to lower energy levels. And they have a lot of energy at the top. So while they're like going down, they're losing energy. And so that's why they're losing it, because they're going down energy levels. So the electrons from NADH can start up here because they have free energy that's around here, but the ones in FADH are just a little bit lower. So that's why they can't enter at the very beginning. It's interesting to note that there's a lot of iron is used in all these complexes. Anyway, yeah, so that's that. That's kind of what I just said. It helps you control the release of the energy from the electron, so it's not a giant fireball. It's a slow, stepwise release of the energy. Down, down, down. And each one of these carriers is more and more electronegative, and you have the most electronegative element at the bottom, which is the oxygen. So they're slowly being pulled down because you're increasing electronegativity as you go down. These are the pictures in your book. Same kind of ones, they're just whatever. Now, we just talked about this over here. And the point was to make this right here, this hydrogen ion gradient. But why do we care? Because hanging out in this inner mitochondrial membrane is another enzyme called ATP synthase. ATP synthase, what can happen is that hydrogen ions will flow through it. And as they flow through, it'll cause it to rotate. I'm going to show you videos on this. Anyways, and it'll cause it to become enzymatically active down here, and it will phosphorylate ADP to become ATP. Okay? So there's some terminology you need to know. Something called chemiosmosis. When you think of osmosis, what do you think about? Diffusion of water. Let's take water out of the equation. Osmosis is just the diffusion of something. Okay? Chemi. Add chemi there. We have the diffusion of an ion. Chemiosmosis is the diffusion of an ion. So it's a complicated term, and you could be sort of intimidated by it. But if you actually think about what it means, it's not that intimidating. Chemiosmosis is diffusion of an ion. In this case, we have diffusion of hydrogen ions. So you can link the electron transport chain to the production of ATP via something called chemiosmosis. It produces the gradient, then hydrogen ions will flow down their gradient via chemiosmosis through ATP synthase, activating it, causing it to make ATP. Okay? The way we made ATP in glycolysis in the Krebs cycle is called substrate level phosphorylation, using an enzyme to directly transfer a phosphate to ADP. The way ATP is made using this whole process is called oxidative phosphorylation. Okay? Because you're not directly taking a phosphate off of another molecule and putting it onto ADP. You're creating this large gradient, allowing it to flow through this enzyme, and then you're making ATP down here by phosphorylating ADP. So this whole thing combined together, this plus this is called oxidative phosphorylation. Ooh. What do you think oxidative is coming from? Why oxidative? What is oxidation? Loss of electrons. What's happening during this whole process? These electron carriers are gaining, but then losing electrons. These things are all slowly becoming oxidized as the electrons are passing through it. Therefore the name. Okay. All right. Okay. We gotta watch the video. So we made up a gradient. We allowed. All right. So then we have to talk about these two things. So the hydrogen's moving through this guy. This is a complicated enzyme. So as they pass through, I'm going to show you some videos. You accumulate a couple of hydrogens, and they cause this whole thing to rotate. Well, this rotates. There's like a knob down here. And as this knob is rotating, these are the catalytic sites on the enzyme. And as the knob is rotating, it's causing these sites to become enzymatically active. So it can therefore phosphorylate ADP to become ATP, okay? The protons moving through this, causing this to turn, is something called the proton motive force. I think it's in your textbook. You need to know that terminology as well. Proton motive force. I think I have it in here. Proton motive force. Oh, there it is. I did put it on.
Oh, wow.
The protons are diffusing down their gradient through ATP synthase. They cause it to rotate, part of the knob on it, and as it's rotating, it's activating these catalytic sites on this part of the enzyme. Oh, I have a number of videos. Okay. The chemiosmosis, I already talked about this. I will send you all these PowerPoints. It's important to know that chemiosmosis is what actually links the ETC to the actual production of ATP. Because the ETC set up the gradient, right? I promise y'all, after tomorrow, this all should start making a lot more sense. Because you're going to be forced to teach it to me. Okay? And by doing so, you're going to have to learn it, yeah.
Like, is the ADP and the phosphate already there?
Yeah, so the ADP and the phosphate, the cell, yes.
It's already there.
It's already going to be in the matrix.
Okay.
The cell uses up ATP very, very rapidly, and it diffuses back
Okay, that's what I
into,
was thinking.
yeah.
For a little bit of clarification, the H plus gradient is an electrochemical gradient.
Yes. Okay. Before we do this, let's watch our videos. Do you want to see generalized video of the whole thing or specifics on the ETC and ATP synthase first? Well, I'm going to show you all. Okay. Actually, no.
Can we just watch Halloween?
What?
I like that. Yeah, I like that. I like that one. Jay, what?
We have to watch Halloween.
Oh, that movie. The Trigger. Oh, that's
You ready?
Yeah.
Yeah, this sounds kind of freaky at the beginning, but it's not.
Halloween, it's okay. Can we watch a Halloween movie?
No.
The electron
transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred to these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome CC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, two mobile carriers are also involved: ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and CI, which combine to form ATP. At the start of the electron transport chain, two electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of one hydrogen ion for each electron. Next, the two electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. One hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with the molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradient animation. This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
Does that help a little? The reason why it said, if anybody wanted to know, in that 4th complex, it talked about there were 4 electrons that were sort of taken together. It's because molecular oxygen, O2, you don't ever want to split it up inside of a cell just to become an O. Those are called free radicals, and they're like Chucky. You know, like the little kid doing out horror films, like crazy little kid that would kill everybody. Okay, like a O by itself is a free radical, creates lots of cellular damage. So cells don't want to form free radicals. So they'll keep the O2 together. So in order to allow that to form water molecules, you have to take the 4 electrons, 4 hydrogen ions, and O2, and they'll split it all up to make 2 waters. Anyways.
Are the hydrogens there, the complexes, or like, the first 2 and 4?
What, Lauren?
Does it ever occur, like, or whatever, where the oxygen
No. I mean, it can happen as a byproduct of metabolism, but that's why you have antioxidants. They help take them out before they become damaged. All right, this video is on ATP synthase itself.
Yeah, for complex 4.
What about complex 1?
Complex 3 is different than complex 1 and 2. It's still like
Concentration gradients are a key component of the biological world. The potential energy from these gradients is often used to perform biological work. Here we will focus on hydrogen ion concentration gradients. Hydrogen ions are also known as protons. A gradient exists when there is a higher concentration of a molecule in one compartment compared to a neighboring compartment. This animation will demonstrate how the potential energy that results from a hydrogen ion gradient uses ADP and inorganic phosphate, also known as PI, to synthesize ATP. This process involves an enzyme complex called ATP synthase. Gradients and the potential energy they create are key aspects of the biological world. A good example of the use of a gradient occurs in the mitochondria when ATP is synthesized. ATP is synthesized by ATP synthase, a large complex of membrane-bound proteins. Here we see ATP synthase, along with other membrane-bound proteins. Notice the large difference in the number of hydrogen ions on the two sides of the membrane. This difference is a hydrogen ion, or proton, concentration gradient. The energy associated with this gradient is used to synthesize ATP from ADP and PI. This occurs at the ATP synthase complex. One hydrogen ion enters the ATP synthase complex from the intermembrane space, and a second hydrogen ion leaves it on the matrix space. The upper part of the ATP synthase complex rotates when a new hydrogen ion enters. Once 3 protons have entered the matrix space, there is enough energy in the ATP synthase complex to synthesize one ATP. In this way, the energy in the hydrogen ion gradient is used to make ATP. Now let's watch the process again. Notice how the proton enters the ATP synthase and exits into the matrix space. Once 3 more hydrogen ions have crossed the membrane, another molecule of ATP will be made. In this example, the hydrogen ion gradient is large enough to produce 6 ATP molecules. Please watch as the remaining ATP molecules are synthesized.
Please watch.
She said please.
Oh, yes, sir. I don't think we complex
The process has now completed, and the result is an equal number of protons on each side of the inner membrane. Without a gradient, there is no more energy available to make ATP. In biological systems, however, a gradient is always maintained. The mitochondrial hydrogen ion gradient is generated as electrons pass through 3 membrane complexes. That process can be seen in the mitochondrial electron transport chain animation.
Do you want to see another one? Or no? No? Yes.
No.
In the mitochondria, the energy stored in NADH is used to generate a proton gradient across the mitochondrial membrane, and the energy of the proton gradient is used to make ATP. When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD and FAD are reduced to NADH and FADH2. Inside the mitochondrial matrix, the electrons from NADH are transferred to the electron carrier coenzyme Q by NADH dehydrogenase, and the protons are transferred across the membrane to the intermembrane space. Coenzyme Q carries the electrons to the cytochrome BC1 complex. As the electrons move from the BC1 to cytochrome C, more protons are carried from the inside to the outside of the membrane. Electrons are also transferred from FADH2 to coenzyme Q, with the protons being transferred across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen. The terminal electron acceptor and water is formed as the product. The transfer of protons to the intermembrane space generates a proton motive force across the inner membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate.
Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
Yep.
So the complex two only, like it releases hydrogen ions and complex two is like what is the complex two do?
Oh, okay. So that video is actually kind of wrong. All it does is take the electrons. It doesn't pump any hydrogen.
Oh, okay.
No. No. And your book talks about it. Like if you actually read, do you remember reading in that part? It talked about the accounting, like how much energy is actually made is a little bit, how much ATP is actually made is a little bit tricky because one, the amount of electrons or the amount of hydrogens pumped by the electrons from FADH2 are different from the ones from NADH because they don't pump at that first one, or where they come in anyway. So yeah, that video is wrong.
Can I get some water?
Go ahead. Yeah.
Just clarification, hydrogens that are like pumped out from complexes 3 and 4, are they like already in the matrix and they're just pumped out? Or like?
Yeah, because they're constantly flowing back down. They're gradient through ATP synthase.
Oh, so they, yeah.
Yeah. It's always going on.
Cycle.
It is. All right, we're not finished. No, what do you think? No. All right, I'm going to ask you. So, Clayton, where does the glucose come from? Clayton? Outside. From where? Where does your glucose come from? Food you eat. Good. It's not hard. These aren't hard. All right, where does the oxygen come from, Sarayu?
Negative pressure from the lungs.
Sarayu? Huh? Where does the oxygen come from? This O2, right here. Yeah. Breathing, or if you're a single cell organism, from just the outside. Good. Where does the CO2 come from, Chris?
CO2 comes from like after you like breathing the oxygen.
Oh, good God, no. Use that we talked about today.
Elevator yeah. It's like that. It's at the end of the Krebs cycle.
Yeah, okay, good. The Krebs cycle and the oxidation step. Yeah.
You make like a lot of glycolysis, right?
No.
That's fermentation.
No, yeah. No CO2s are made in glycolysis, the oxidation step in the Krebs cycle. Y'all have to pay attention. Where does the CO2 go, Chris? You can answer that one. Good. It diffuses out of the cell. Good. Where does the water come from, Leah?
From the oxygen binds with the hydrogen ions and the electrons.
Perfect. Jazzy, where does the ATP come from? This is a multi-step answer.
ATP synthase.
And this is the whole of cell respiration.
It's also from when you turn from glycolysis and from the Krebs cycle.
Good. So glycolysis in Krebs is called substrate level phosphorylation. Most of it comes from the oxidative phosphorylation, which is ATP plus, ETC plus chemiosmosis. Okay. What else is produced that's not listed? Clayton. Okay, good. Yeah. Yep. Because they're used. What else? What else?
Heat energy.
Heat. A lot of heat energy is produced. So why do we breathe?
Because we need the oxygen.
Because we're aerobic.
We're actually always aerobe, so we need oxygen.
Taking in oxygen, but also getting rid of
CO2.
CO2. All right, so let's talk about this. So the final electron acceptor is, everybody?
Oxygen.
Good. So what happens if there's no oxygen?
We die.
Glycolysis.
Let's follow me. If there's no oxygen, the electron transport chain gets backed up. Let's say that there's a giant
There's a bunch of electrons in complex.
car wreck where no one's injured on the interstate, but it's like horrible, like a semi like dumped all stuff everywhere, but no one's hurt. Okay, anyway. So all of a sudden things get backed up, right? Like even like stuff gets all backed up, people can't even get off the exits. It's like it gets all backed up. So the ETC gets all backed up if there's no oxygen. The electrons get stuck. there's nowhere for them to go, okay? And then if they get stuck, then NADH and FADH2, like they can't get off on they can't drop their little passengers off. So then you can't regenerate NAD plus or FADH2. So then what stops? Glycolysis or the Krebs cycle, peroxide oxidation stuff, all that stops. So then we stop making ATP, and then you run out of energy, and then you die. Yeah.
What is cyanide? You never taught us like what cyanide does with my
I set it up for you. Cyanide.
Oh, hey.
This might help you, right? Is this one of your questions?
Yeah.
All right. So cyanide is now this figure showing what different poisons will actually do to you. But cyanide blocks the cytochrome complex 4, prevents it from functioning. Okay. So if you can't work here, then it's basically the same thing. Like everything gets all backed up, and then you can't make your ATP and all that, and you die. Yes.
So back with the backing up of the ETC and everything, like you don't have oxygen, why doesn't that just like straight go into fermentation?
The only cells in your body that undergo lactic acid fermentation are your skeletal muscle cells.
Right.
And even then, they won't make enough to last very long.
Yeah.
So, yeah, okay. We're not finished. Don't get your hopes up. Oh, we only have that's okay. That's okay. We need to talk more. No. I'm not talking about all this. Okay, so, no. All right, we can use a lot, I'm only using the figures here. We can use a lot of other food sources besides straight glucose. I talked about this at the very beginning, right? I said we talked about glucose because it starts at the beginning. You don't have to do anything to it. But if we needed to use proteins, other carbohydrates, or fats, you can, but things have to happen to them before they can enter. All right, so sugars, depending on what they are, they can be modified to become straight glucose and enter right at the beginning. Sometimes the sugars have to be broken down if they're like polysaccharides or whatever. All right, fat. You can break off the fatty acid tails and then use the glycerol. You can modify it a little bit to enter right at the second step to become G through T.
What do we do with the fatty acid tails?
We'll talk about those in a minute. They'll become acetyl-CoA.
Wait, I thought G3P was like guanine.
No. Glycerol 3-phosphate. G through B. The amino acids, you take off that amine group, you can actually produce a pyruvate molecule, and then they can enter in there. They can also become acetyl-CoA. Now, the fatty acid, they can all, you like cut them up and they all become acetyl-CoA groups. You can make 1 million acetyl-CoAs from like one fatty acid tail.
So that's why it's like
Yeah, that's why they're so dense in terms of amount of energy in one fat. You can make a ton of them. You can take 2 carbons, think if they're like 20 carbons long and there's 3 fatty acid tails, you can make like 10 acetyl-CoAs from one fatty acid. So then like 30.
So when we use proteins, we spell that as urea?
Yeah. Yeah, urea. Well, you convert the NH3 to urea. Anyways, okay. I'll stop tomorrow. I'm going to start at the beginning talking about this a little bit, and then we'll do our drawings. Your essays are going to be due on Wednesday.
Can you just put this back in our folder if you want to do the correction?
Yeah. Yeah. Yeah, we have a research meeting, but yeah. Yeah.
I have my paper, but like sometime between last week and this week when I was running my paper, my materials section got completely deleted.
Oh no.
So yeah, you want me to turn this in and bring my materials tomorrow, or just turn it in all at once?
Give that to me. I'm going to turn the rest of them have it. Thank you.
It's just like somehow got cut off. It's my revision still.
It's okay. Okay. It's fine. It's fine.